Yes.
Yeah, good, thank you. Yeah. Yeah, thank you for the opportunity to present this session. I am a plant breeder at University of Sydney. And today I'm going to talk about designing and conducting field experiments in agriculture. And I don't want to pretend that I am an expert in this area, but I'll try what I have learned over the years. Uh, yeah, I have been working here in the field of agriculture for nearly 30 years and about 25 years as a plant breeder in Australia. And I used to be a plant breeder in Nepal as well. I used to work for NAC. I was a wheat breeder at Bhairava, National Wheat Development Program. That's what they used to call before. I think it's still called now. All right, so let's start the formal session, and we we want to test something. That's why we needed experiment, and we would have some hypotheses to test for, right? So a hypothesis could be varietal differences exist among test lines, for example. Yeah, optimum sowing time is first week in April, or 100 kg nitrogen gives highest yield in rice or wheat. And this kind of things you have as a hypothesis. But you need to test those hypotheses. That's why you need some experiments. And objectives can be a single objective or can be multiple objectives, depending upon what kind of experiments you are doing. And variables, yeah, you need to find what variables you want to measure. For example, it could be a grain yield or plant height, plant biomass, flowering time, maturity, disease resistant, or anything. But you need to have those already defined. What are the variables and how you are going to measure them. And also you need to do the define the treatments. And what do you mean by treatment? Treatment is anything that you are going to test. For example, if you are going to test uh, different varieties, you want to know which is the highest yielding variety, then your variety is a treatment. Yeah, that's how we define treatment in the experiment. Then you have to assign those treatments to experimental units, and the experimental units can be field plots, or it can, if it is a glasshouse experiment, it can be pot. And then you have to assign those treatments to experimental units. And then you take the samples, that's what is data. You collect lots of data, then you analyze the data, and you write a report, then you publish. That's the main thing you want to do. But today I'm not talking about these two topics highlighted in yellows. They will be covered in separate topic, but data analysis I think is already covered yesterday, and writing and publishing will be covered today by Dr. Jagdish Tennyson. And field pulse designs, the main thing is the as I said before, hypothesis. We have a hypothesis, but you need to make sure the hypothesis is realistic, achievable, measurable, and time bound. It can't be just opening it. Oh, I want to do this experiment, but you need to know why you want to do this experiment and how long does it take to do it, what resources you have. Yeah, that kind of things, it need to be predetermined. And objectives has to be clear. It has to be measurable, and it can be a single objective or can have multiple objectives. In when you want to test something, there are some fundamental things in the methodology. What are they? I call them R square C. So replication, randomization, and control. These things are very important, and you should not forget them. And why do you need replication? Why do you need replication? Yeah, to estimate experimental error. So if you cannot or experiment, then it doesn't have any replication, then it's not valid because you cannot test properly. It doesn't have the error component variance. And same thing is randomization. If you don't randomize your treatments, then you'll be biased. Then you need control. Other thing is control. Why do you need control? Because you want to compare your results to measure the progress. So whether you are doing any better than, for example, if you are testing different varieties and you want to know whether some varieties are giving higher yielding than your controls, because that's what you want. You don't want to have a variety that is lower yielding than control or same yielding as control. You don't want that. You want a better than what you already have. So that's control. That's why we must have a control for any experiment we do. And other thing is location of experimental sites. So what you want to do experiment now, you have defined objectives, you know what you are going to measure. Now what you need to do is you need to find a place where you can do your experiment. It should be large enough to accommodate experiment, minimum variability, that is soil type, nutrients, slope. Yeah, there can be many different things. So it has to have a minimum variation of those things in the field. And you need to know the crop history as well, because you don't want to plant a trial where there was a chemical. Hello. Okay. So yeah, there is some background noise. Could you please keep it mute your microphone?
Yes, I have put on mute.
Yeah. Thank you. Yeah. If you want to ask me question, you can do so by unmuting. Otherwise, I suggest you keep it muted, so we don't get that noise in the background. Yeah. And because what I was saying is crop history is a very important. You don't want to have a trial where your trial would be compromised by previous crops history. For example, if there was some herbicides applied which is not compatible to your crop you're going to do, and the herbicide has a residual effect and it will destroy your trial. So you don't want to happen that. Yeah, this is kind of, common sense things, so I don't need to talk about too much about these things. The other things to consider while doing an experiment is to minimize the variation. What you want to do is you want to test something so that only that particular component is variable, and all other treatments you have is constant. Yeah, you need to make that sure. To do that, the technique we have, one is blocking. Why we need blocking? Can anybody tell me? All right. Well, I have an answer here already anyway. Yeah, blocking, it's avoid. It doesn't avoid, but I'd say sometimes it can avoid, but reduce variability. For example, if there is a soil type variabilities in the field, yeah, sometimes you have a bit of difference in soil types, some a little bit sandy, some a little bit clay can be. Yeah, so that kind of things, if it is there, you can minimize that by blocking. And if there is soil fertility level variance, which you know by previous history, then you can avoid that by blocking. And the other thing is plot size. A plot size would depend upon the objectives of the experiment, land availability, and resources, what kind of resources you have, and also whether you are going to plant or harvest with the machine, or you are going to use manual labor. I believe in most of the cases in Nepal, yeah, there will be manual labor used, which is a very easy to control, and you would have a very minimum error. Because I used to do that sort of experiments in Nepal, and you could control most of the variables easily. But if it is a machine planted, machine can do anything. So sometimes they are perfect, sometimes they are not. And also the border effect. You have to think about how you can minimize the effect, aging effect, and the border effect. Sometimes you can do that by trimming edges or taking samples from the center plot. But if you do that, then your plot size would be reduced. So the smaller the plot size, the accuracy would be smaller as well. So you need to think about how you can avoid the border effect. So yeah, just why we need arrow techniques. These are all techniques you are using is to reduce the variation in the experimental, to minimize the experimental error. So what you are measuring is the real differences, not the environmental differences. Yeah, that's your main goal. Mm. Yeah, yeah. I just realized that I didn't have the display of slides. I just noticed that now. Okay, and in Australia, this is the plot planter. Yeah, and all the experiments we here are planted by a machine. See, it has a 3-layer, see, 1, 2, 3. So it can show 3 rows at a time. And the person here is sitting and it pours the seed and they have marked how long and the tractor has to drive before changing the seed. And this is how they can plant. And don't think we do only the machine planting. We do hand planting as well, just like in Nepal. So we see this is in small area is a board cage, and here bringing big machine is difficult, so we do by hand as well. And this is just to show that on my left, this is how the trial looks like in the beginning, and this is at later stage. And this is an aerial photo of the trial plot. See, you can see the edge. You can see this edges here, and this is the plot. Yeah, this is the plot. So you can easily see how the plots are laid. And these are all machine planted. And we have here about 4 hectares of trial land here. Okay, so types of experimental design. Now we got some ideas about how we can go for designing a trial. First, we have the objectives, hypotheses we have, and we have the experimental sites, and we know the blocking and other ideas about minimizing the variation. So what kind of design you want to do a particular trial? So it will depend upon what kind of trial you have. What do you want to measure? The nature of the trial. Yes, single factor experiments where only a single factor is involved, such as grain yield. You want to measure the grain yield. So in that sort of experiment, it can be done as a completely randomized design. Yeah, completely randomized design is as the name suggests, it's assigned, treatments are assigned randomly. And no blocking. There is no blocking. We were talking about the blocking before to minimize the variation, but there is no blocking in this kind of experiment. Yeah. And replication, it can be replicated, but replication also is random. There is replication, it means repetition of the same treatment. Huh? So it's not in a block. Yeah, there can be 2 or 3 repetitions, but they are not in a systematic blocks. They can be side by side as well, randomly. And this kind of experiments is applicable only in ideal process where there is no variability. The soil is where the soil is very uniform and there are no external factors, which is hardly correct, right? And that's why it is not used in the field experiment. Yeah. Why? Because there is no such a thing of not having a variability in the field. There is a soil variability, there is a fertility variability, yeah, there is an age effect. There are so many variations there. So we normally don't have this kind of design in the field because we cannot control the variation. And other method is randomized complete block design. This is the design most people use it in agriculture, and here we do the blocking. Blocking removes or minimizes the variation, as we talked before. But it does only one direction. Unidirectional variation is controlled, because if the experiments are, if the variation is, for example, north to south, according north to south, then it will you can block from north to south. So if it is going north to south, then you can block from east to west. So the variation within a block is small, but between the block becomes big. That's how you do the blocking. That's the main idea of blocking. Minimum variation within the block, but maximum variation between the blocks. That's what you want. And every block contains all treatments. So if you have 20 treatments, then in one block, all 20 treatments are planted in random. It's a full replication. And the other method is a Latin square design. The randomized complete block design is useful when the variation is unidirectional. But what are you going to do if you have land and that has variation going in two different directions, perpendicular to each other? So comes the Latin square design. We can do that because in this method, row and column blocking, you can do row and column blocking, and that minimize the variation between the rows and between the within the row and within the block. And the drawback, what is the drawback of this kind of experiment? Same number of rows and columns are needed. And so you need as many replication as the treatments. If you have 20, for example, 20 treatments, then you need 20 replications, which is kind of impossible to have in the field, right? So it will be a good experiment. You have very few treatments, okay, 5. So you can have 5 treatments and 5 replication, yeah, 25 plots. Yeah, that's doable. But if he has 20 and 20 times 2,400 plots, you need a huge land for that. Yes, as I said before, this is a 4x4. This example 4x4 lattice square design. You can see here A, C, B, D. See there. See, every row has a treatment just once, and same thing. Every column has a treatment just once. Yeah, you can see that. And here, what it is showing is the fertility gradient is going this way, and the soil variation gradient is going this way. So you are blocking here like this. See? But as I said, this becomes too big to handle when there are large number of treatments. Can you now tell me how many reps are in this experiment? Hello? Hello?
Yeah, 4
Yes.
applications.
Yeah, thank you. Yeah. Yeah. So this 4 treatments, that means 4 applications. Yes. Yeah, that's the drawback of this experiment. You need as many replication as treatments. And then it is not possible to do that kind of experiment because it's too big. So then it comes to incomplete block designs. And incomplete block designs, there can be a different variation in incomplete block designs. I'll outline only couple here. What is lattice design? And here it is good design, but what happens is the number of treatments must be perfect square. For example, 16, 25, 36, 49, 64, like that. And this is okay, but because of this requirement of the the square root issue, and this is not used very commonly. Sometimes people use it for particular reasons, but not very frequently used. And in lattice design also, you can have a balanced lattice and a partial balanced lattice. And in the balanced lattice, the block size should be square root of treatments here, just as we said, because perfectly square. So if it is 36, then the block size should be 6. And replication is need to be +1. So if it is 6, it will be 7. And this becomes too big to handle. And also it is complicated. The analysis becomes very complicated. Yeah, that's the other reason why people don't want to use it. The partially balanced lattice, sometimes they use because you don't need as many replication as needed as in balanced lattice design. So to compromise that, they came with a partially balanced lattice design. Yeah. And this is also not very common practice. Yeah, and the other design is limited or no replication. Sometimes you have some issue with the land or with the seed, then you can't have a replication. And also there can be sometimes the number of lines you are going to measure are too many. As a plant breeder, I frequently do this kind of design. Here, what we do is we test so many. We test about 2,000 lines. Okay, think about it. When I have 2,000 lines to test, and if I want a replication, so 2,000 two replication will be 4,000 plots, which is too many. Huh? And this becomes too big to handle. It needs a lot of resources, and also at the same time, this kind of experiment is useful in the very early stage of the varietal development. So, for example, you are getting seed from a single plant selection. So you have only a handful of seed. You can't replicate it anywhere. Yeah, that's so. The need for this kind of experiment came from two reasons. One is too many lines to test. And other one is you don't have enough seed to replicate. So in that case, what we do is we just use a single rep. But how do we test many lines in a single rep? Because we have multiple checks. Yeah, I'll show you here. See? Yeah, yeah, we divide the land into different blocks. Block 1, block 2, block 3, for example. See? And I have here different controls. See, control 1, 2, 3, 4, randomized in every block. Yeah, we have controls. And in the rest of the area, rest of the plots, we have the different lines we want to test. So what we do is we can use this as an error variance to test the mean of different lines. Okay. So difference between checks and nutrient. Then we can compare the means of different lines with our controls. Yeah, you can also compare differences among new treatments because you have the error variance there. So from that, you can measure this least significant difference. I think you guys were taught yesterday about the list, data analysis. So yes, single list differences you can measure. Then you can measure same thing by different among check treatments and treatments. So all these you can measure in this augmented design. This is very useful for early generation line, very early generation line where you have a very limited seed and the number of lines you need to test is big. Then under comes the partial rep or P-rep design. Because this type of design is used when the seed amount is low again to applicate, but the requirement of this type of design is you should have some entries, some lines with enough seed so you can replicate at least twice. As a rule of thumb, they said about 25% of the lines should be applicated in this kind of experiment. Yeah, but sometimes we don't have even that sort of seed, so we even go for low number of repetition. In that case, we compromise by including more controls and also more reps in the control. And this is a very commonly used in plant breeding experiments. Yeah, so, you know, what happens in the plant breeding experiment? First, you have that augmented design from 2000, and you come to about maybe 700 or 800 lines you reduce to. Then comes to period design, and you do this kind of experiment. This is will be kind of a second year of experiment for field evaluation. This kind of experiment. And here, this is just an example. I just copied from the website. Here it is the link. And what it is showing is that the green plots are replicated. Here it is about 25% plots are replicated and others are not replicated. To analyze the data, now we have different types of softwares. You can get them sometimes freely available as well. For example, R, you might be familiar with the R's, and it is free software. Anybody can use it if they know how to use it. And also there are lots of other softwares which you need to pay. Okay, and we talked about the one factor experiment until now. What we are interested in measuring one factor, and I was giving as a grain yield as one factor. But now, in fact, when it comes to when you have to test more than one factor, we call that factorial experiments. And these should be independent variables. It can be 2, 3 factors. For example, if you want to test 3 herbicides and on 5 varieties of wheat, 3 different types of herbicide, and 5 different type of wheat. That is 3 by 5 factorial, so it will be 15 treatments. And if you have 4 reps, then you can have 60 plus, which is manageable. And 2 levels of seeding depth on 6 varieties of wheat, 2 by 6 at 12 treatments, again, if 4 reps, 48 plus, easily manageable, and in this type of replication design, what you can get is a main effect as well as interaction effect of both factors. And it can be laid out as the number of treatments are relatively small, as we outlined before. But think about the complexity of the trial. If it involves some mechanical issues, and if it is herbicide trial, the spray drift from one herbicide to another, from one plot to another, right? And is it possible to do that? Yes, so you need to think about this. Can you do this kind of experiment in the RCBD? If the treatment factor, the rule is if the treatment factors cannot be precisely controlled, then you should not be doing RCBD. You should do something else. Okay. So what you can do is here. This is just example of factory experiment. So you have 3 rates of nitrogen, 5 varieties of wheat, 15 plots. Right, so you just did it in 1 rep. You can do the same thing in 2nd rep, 3rd rep, 4th rep. So it is possible because the fertilizer rate it can control within the plot. So it is easy to do that, and also varieties you can control. But if you want to apply different herbicides, in a small plot, and also you want to change the seeding depth of planting each plot will be very difficult task. So how you can do that? Then start experiments. We can do the split plot designs. It becomes easy to manage. What you can do here, I have given here see 3 types of herbicides and on 5 varieties of wheat you wanna do. So here herbicide 1, you can go this way, so in this block you get all the same herbicides. Here a different type of herbicide, the same herbicide, and same thing here, different herbicide, but different varieties of wheat. So what you do is you can have a replication, 3, 4, as many replication as you can handle. So it is doable in this kind of experiments. So what you are trying to do is, if you were using different in factorial experiment, RCBD, then you would have to apply different fertilizer, herbicide here, and different herbicide here, different herbicide here, which is not possible because you will have a spray drift, right? But if you use the same herbicide, you just go like this with the machine and use the same herbicide all the way, right? So it is possible. So that's why this kind of trial has been designed. Split plot system and design. And sometimes we want to add a third factor. Okay, and if you want to give a third factor, then we call that split plot design. So first factor is in the main plot, and then subplot is the second factor, and the subplot goes the third factor. Yeah. This and this kind of in this kind of experiment, what happens is how you allocate the main plot and subplot will depend upon the objective of the experiment. For example, if plant breeder wants to test different herbicides on different varieties of wheat, because he will be emphasizing more on the variety. So in these plots, he will assign varieties in these plots, and he will have the herbicide going this way in the main plot. Yeah, this is main plot, this is main plot, this is main plot. Okay, and the little ones are subplot. So, but if an agronomist want, in his case, herbicide effect would be probably more important than the varietal differences. So he might do the other way. He might do, okay, no, I want to use just one variety and different rates of herbicide. So he can go the other way. So it will depend upon the objective of the experiment. And you would have heard about the nested designs as well. And yeah, the nested or hierarchical designs, it is different from split plot, as the treatments are not identical. And this is again, this nested design is very complicated. It can be a complicated design. It is not normally used in agriculture, but it is used in different type of experiments. For example, I have given you the experiments involving schools and teachers. If somebody wants to know the performance of a math teacher in different schools, yeah, they could use this experiment, because teachers are nested within a school, yeah? Because in one school there will be teachers, different teachers, but all teachers and schools are different. And the other example is leaves are nested within a tree. So if somebody wants to test the different performance of different leaves on different trees, and leaves and the trees are different things, because you are not testing the same thing all the time. Like in split plot design, what do? You are testing the same varieties all the time, but here not, because the leaves are different, trees are different. So it is a complicated experimental design. I just wanted to give you some just some examples, and I'm not going to talk about this anymore. The split plot designs, I already said that when the third factor is needed, that we can use the split plot design. It is similar to the split plot, but it is third factor, it is split. And split plot design, yeah, this is also another design which we don't use regularly. And in split plot design, we split the plot into main plot into split. Here we do that in strips. Here the objective is measure the interaction between those two factors. The interaction factor is more important than the individual factor. Yeah, that's why you do this kind of experiment. And this, for example, yeah, well, I've just given 3 levels of spacing between rows and 4 levels of sowing depth need to be tested, as both factors will need large plots. A split plot will not be practical. Okay? Because changing the different depth and spacing for each plot would be difficult. People use this strip plot designer for that kind of experiment. All right, so what we have now, we are I have explained a few things on the experimental design. I hope I gave you some idea about the experimental design. It's not the complete explanation of things because we don't have enough time to explain everything. This is just kind of introduction. I know you would have studied this in your experiment in your somebody's okay, yeah. You'd have studied this in your undergraduate or postgraduate studies. So you are already familiar with this. This is just a kind of a refreshing. And you can get a lot of information these days from the internet. Yeah, you refer to the internet. Yeah. So just a few questions. What are the most commonly used field designs in agriculture? Which one do you think is the most commonly used design in agriculture?
You can use chat room as well to provide the answers so that we can discuss.
That's right. Yeah. And yeah, something like why do we need blocking? Yeah, blocking is I think I explained already. Blocking is done to minimize the environmental variation. Yeah. And why is randomization needed? Yeah, if it is not randomized, then it will be biased. Okay, the human eyes are biased. So if you don't randomize, the experiment has no validity. And when is a split plot design is used, and this it will be used when you have to have test two factors, and also the precision for one factor is more important than others. Okay, yeah. So you use split plot design in that case. And how do you minimize the two-way variation? I think I have explained that as well in the Latin square design, because you have the row blocking and the column blocking, so you can minimize two-way variation in that. Okay, now you have the experiments, you design the experiment and you planted the experiment. So what do you want to do next? Next thing you want to do is you want to take samples from your experiments, and sample taking is measuring the variability. How you can measure, for example, you have a trial and you want to use test the measure the plant height of different lines. You have 100 lines and you want to see the plant height for all those 100 lines. So what do? You go to the, you go in the plot, and is it possible to measure all the plants in a plot? Obviously not. So what do you have to do? You have to take samples. Huh? Here I'm just giving you one population. What is population? Population is a big thing. So population, for example, in a plot can be all the plants in a plot is a population, right? And sample, it can be a single plant or a portion of the plot. Sometimes people use quadrant. So it's about half 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 of half square meter, some frame, some sort of frame. They just throw it in the plot, and wherever it lands in the plot, and they take the measurement in that area. So that is a less bias. Yeah. So yeah, that and sometimes when you use human eye to pick up the plants, you are always biased because human eyes are biased. You want to go with the good looking plants, and so sometimes and then people say, oh yeah, I only take good looking plants, I should take the better one as well, and then probably then you go only for the balance. So it's always biased there. So to avoid that biasness, you need to have some method of sampling. Sample size will depend upon the variability in the population, and what the main thing of taking sample is it should be representative of the population. Okay? If you are taking, for example, the weight of say 100 seed weight, you took yield from several hundred lines, and you want to know what is the 100 seed weight of different lines. So what do? You take a sample of this from the seed, just a handful of grains, and hopefully there are 100 grams, 100 grams, at least 100 grams. Then you count those grains and take the weight of that. But if you just pick that by hand, yeah, 1, 2, 3 like that, you will always pick the big grains. So don't do that. You should just take a sample and put it in your seed counter, and it will sort it out itself. Yeah, and it will give you the number. And just need to remember that human eyes are always biased. So just don't believe in your eyes while taking those sort of samples. Yeah. Well, I think I have covered most of the things I wanted to cover today. Are there any queries or questions? Hello?
Hello. Yes.
Yeah, any queries, questions?
Please put your questions if you are if you can in the chat box as well. I can pass it to the speaker.
All right. Yeah. So yeah, looks like there are no more. There are no questions. So yeah, hopefully we're.
Hello, sir. Namaskar.
Yeah. Please go ahead.
Yeah, सर यो प्लान्ट ब्रिडिङको अलि लेटर स्टेजको रिसर्चहरूमा
हजुर
जुन एग्रोनोमीको ट्रायलभन्दा एक्सटेन्सन गर्नुभन्दा पहिलाको स्टेजमा जुन बेलामा चाहिँ मल्टी लोकेसन ट्रायल भइरहेको हुन्छ
हजुर
त्यसमा चाहिँ हामीसँग इनफ सरप्लस सिड हुन्छ र सँगसँगै हामीलाई त्यो सिड चाहिँ सिड प्रोडक्सनको लागि पनि चाहिन्छ र ट्रायलको लागि पनि चाहिन्छ। यो दुईटै पर्पोजको लागि गर्न मिल्ने खाले रिसर्च ट्रायल डिजाइन चाहिँ के होला सर?
It depends upon the purity. How much purity you want to have in your seed or seed production. Okay. For cross-pollinated crops, if it is a cross-pollinated crop, like for example, maize, then you don't want that seed to be reused, okay, because it is all cross-pollinated, coming from another plot, so you are not sure you will have the same effect next year. But if you have wheat or rice, yeah, you can do this, and if you minimize any contamination in the harvesting and planting, yeah, definitely you can use the same seed what you are getting from the trial next year. And the design, yeah, the design is I think I would go for this RCBD design, because you are just measuring one variable, you are measuring yield, right? So yeah, the RCBD would be the best one to do. It's less complicated, and also you don't have many varieties at that stage. Maybe you have 25 genotypes and 3 or 4 reps. Yeah, you don't need to have even 4 reps. You can have 3 reps and 75 plots. You can manage that easily. Yeah, did I answer that question?
Yes, sir, got it.
Yeah, thank you. Yeah.
Thank you, Santoshji, for the questions. Any other questions?
Yeah, I can. Yeah. I can stop sharing. And
If you don't have any questions, we can now thank to the speaker, Dr. Kedar Adhikari, for his presentation on designing and conducting field experiments. It is a very big topic. We just got the overview of field designs and some features of main designs that are commonly used in the field that he explained. And as I mentioned, it is just the beginning of collaboration. You can ask when you are conducting experiments and you want to discuss something technical, something new, you can always contact us and we can directly the relevant expert.
Happy, yeah, I'm quite happy to answer any question at the time. Okay, and if you have some queries, and you can email me, and I can yeah, hopefully answer your questions. But don't think that I'm an expert. As I said before, I'm not an expert in statistics. I know only 3 things, but I'm experienced too. Okay. So don't think, oh, Kedar knows everything, so I can get information from him. Please don't think that way. Yeah, I'm not.
Thank you for your humility. And now, we also.
Excuse me, सर एउटा सानो क्वेरी चाहिँ म राख्न चाहे सर। यो जस्तै म प्याथोलोजीको फ्याकल्टी हो सर। अब हामीले सिआरडीमा चाहिँ ल्याब एक्सपेरिमेन्ट गर्दाखेरि मैले एउटा जस्तै अब हामीले चाहिँ फर एक्जाम्पल 3वटा फङ्गिसाइडको चाहिँ 3टा लेभल अफ 3टा डोजमा चाहिँ हामी चाहिँ हेर्दैछौँ कुनै फङ्गसको चाहिँ अब इनिबिसन हेर्दैछौँ सर। भने त्यस्तो केसमा हामीसँग चाहिँ अब त्यो केस भनेको 3टा फङ्गिसाइड छ प्लस 3टा सेम डोज छ। फर एक्जाम्पल 100 ppm 200 ppm 300 ppm छ भने अब हामीले यसो विचार गर्दाखेरि चाहिँ त्यो चाहिँ अब 2 फ्याक्टोरियलमा हैन चाहिँ राख्दा बेटर होला जस्तो चाहिँ फिल हुन्छ। तर त्यसैलाई चाहिँ धेरै जसो कन्डिसन्समा चाहिँ अब त्यसलाई ट्रिटमेन्ट 1 एउटा केमिकल नम्बर के अरे केमिकल एक्स को चाहिँ 100 ppm लाई ट्रिटमेन्ट 1 केमिकल एक्स को 200 ppm लाई ट्रिटमेन्ट 2 गरेर पनि गरेको भेटिएको छ सर। धेरै ठाउँ त्यसरी पनि गर्नुहुन्छ। त्यो भन्दाखेरि त्यो चाहिँ इज इट अप टु दि रिसर्चर त्यो डिजाइन त्यो त्यसलाई 2 फ्याक्टोरियलमा ढाल्ने कि अथवा चाहिँ सिंगल फ्याक्टोरियलै राख्ने कि भन्नेमा चाहिँ त्यो रिसर्चरको चाहिँ सब त्यसमा चाहिँ कन्ट्रोल हुने हो कि के हो सर? त्यो चाहिँ अलिकति
You're using different dose of the same chemical, isn't it?
no, sir. There are 3 chemicals used, and all of those 3 chemicals are applied or are tested at 3 same doses.
Okay, at the same dose. So
Yes.
you have only one factor, isn't it? If you had 3 doses of those chemicals,
Yes.
yeah, 3 different chemicals and 3 doses in each chemical, then that would be a factorial. But if you have only 1 dose, it is just like
what I understood was Santushi was mentioning 3 different chemicals with 3 different doses. Isn't that correct?
Yes, 3 different chemicals and 3 different doses.
Oh, 3 different. Then you have two factors. Yeah. Chemical is one factor, and the other is the dose. Yeah.
Yes. So
And-
are we, sir?
and
So are we supposed to go with the factorial design to factorial?
Yeah, you can do. Yeah, you can do factorial design, and in the lab, because it's CRD is also is very good because you don't have the environment variation as you get that in the field.
Yes.
So you can do CRD as well. So it will depend upon what kind of precision you want to do, and also how many replications you want to do. Yeah.
Sir, but when we go through literatures, when we go through articles, it is often found that scientific researchers are using single factorial CRD in such case. That is, for chemical X, the first dose of chemical X is considered to be treatment 1, and second dose of the same chemical treatment 2, third dose of same chemical treatment 3, and same wise for other chemicals as well. A single dose is comprised as a treatment. So that I've found in many places that such experiments are conducted in single factorial CRD design. So is it is it okay to go with single factorial CRD in such case, or we are obliged to go with 2 factorial CRD in such case?
I think either design is okay, because as long as you do the proper replication, yeah, I think either design would be okay. Yeah. Because you are doing that in the lab condition, and in the pathological experiment, if it is, for example, a glass house experiment, then there will be environmental variation, but a lot of environmental variation. But in the lab, I don't think there will be a lot of environmental variation there. Yeah. Unless you put some prejudices very close to the air conditioner, and they could be cool there. Yeah, only that kind of things I can think about. Yeah.
Okay, thank you. There is one more question, maybe very similar. Is there any variation between factorial CRD and CRD for data analysis? I think it's similar question.
Yeah, it is factorial CRD. So factorial CRD
and CRD, between factorial CRD and CRD.
Well, CRD can be factorial. The difference between CRD and the blocking is in the RCBD. In the RCBD, there is blocking, right? In CRD, there is no blocking, but you can have a replication there. Yeah, so it is just what is it? you are assuming that there is no variation, environmental variation, yeah, where you are conducting the trial. So yeah, they are the same. Yeah. It's
Yeah, I think it is, he wants to know about the ANOVA table design for those calculating error degree of variance. And Naren Dakalji has added another question.
Yeah, well, if you do a factorial experiment in RCBD, then you will have a block as a source of variation block. But if you do in CRE, you will not have a block as a source of variation. Yeah, that'll be a difference. Yeah.
Yeah, that will be the difference to calculate the error variance.
Yeah, that's right. Yeah.
I think this answers his questions as well. So if no more questions, we can move to another session. Thank you very much, Dr. Adhikari, for your good presentation.